All right, we're going to start a new unit, and it is on mitosis and meiosis. And so we're going to start out with mitosis first and the whole cell cycle. The first learning target is students will be able to explain the relationship between DNA and chromosomes in the passing on of traits from parent to offspring. So we're really going to look at what the chromosome structure is, as well as what the whole cell cycle is how those chromosomes get passed down. So the objectives for this particular PowerPoint are to identify and explain the purpose of the 3 parts of the cell cycle. We're also going to identify and explain the purpose of the 3 stages of interphase, okay? And then identify and explain the different parts of a chromosome. And finally, differentiate between a replicated and unreplicated chromosome. All right, so we're going to start with the cell cycle first. And here's just kind of a picture of it off to the right here. But when we talk about the cell cycle, we're talking about the life cycle of the cell. From the time the cell divides and it forms a new one, all the way till it divides and forms a new one after that, okay? And it's the regular pattern of growth, DNA replication, and cell division. All of that is included in the cell cycle. And all of these different phases are included in the cell cycle, which we will get to here in just a minute. So there are 3 stages to the cell cycle. The first one is called interphase. And this is where the DNA, or the chromosomes, are copied, and there's lots of growth, okay? But the main thing is that the DNA gets copied. That's the main thing that happens in interphase. And if you look at this, see how interphase comes right along here? That means that from here all the way to here is interphase. So most of the cell cycle is spent in interphase. The actual process of mitosis is when the nucleus divides. So all the contents of the nucleus divides during mitosis. That's just this right here, which includes prophase, metaphase, anaphase, and telophase. Okay? The final stage of the cell cycle is what we call cytokinesis, and this is when the cytoplasm divides. So that comes right after mitosis. All right? So we go from interphase to the 4 phases of mitosis, finally to cytokinesis. And once that cytoplasm divides, you've now divided that one cell into 2. And that encompasses the whole cell cycle. So let's start first back with interphase. There are 3 stages of interphase. The first one is called gap 1 or G1. That's where there's a lot of growth. So sometimes if you just think of the G standing for growth, that really tells you what happens during those gap phases. This is where all your different organelles get copied. There are some different checkpoints. This is where the cell's going to decide if it's going to continue on with cell division, or maybe it's going to go into another phase where it just kind of remains dormant for a while. The next stage, which is very, very important, is the synthesis phase. This is where DNA replication occurs. We talked about that during the DNA unit. So this is where your chromosomes get copied. And this is going to be important. Remember I told you that every time a cell's going to get ready to divide, DNA replication has to take place because that new cell needs a copy of the DNA, because that's what tells it how to make its proteins and how the cell's going to function. The third and final stage of interphase is gap 2. And this is where there's more growth and, again, more critical checkpoints, because this is where the cell is going to kind of check to see, okay, did that DNA get copied properly? You know, if there's some mutations that are in there, maybe the cell's just going to, kind of tell itself to just die at that point and not go on anymore, so that that faulty cell will not keep getting replicated. So lots of different checkpoints along the way that we'll check to see to make sure that that cell has the DNA that it needs, those chromosomes, and that those chromosomes were copied properly. If you look at what the cell looks like down here, there's not any distinct chromosomes. You can't really see these nice thickened chromosomes because at this time the genetic material is called chromatin. This is like a bunch of unwound DNA. So it's not tightly wound up into these chromosomes that you can see, but it's all unwound. It's almost like, you know, a plate of spaghetti or something. And all those spaghetti noodles are the unwound DNA. When we look at chromosome structure, there's some different terms that you need to know. First of all, chromosome is just one continuous thread of DNA. And then that DNA gets wrapped around proteins called histones. And those proteins influence gene expression. Those proteins, if they're wound up really tightly, will maybe prevent a certain gene from being expressed so that that protein doesn't get made. But then sometimes if it gets kind of unwound and loosened up and that histone moves away from the DNA, then that particular section of DNA does get expressed, you know, transcribed and translated into a protein. So histone proteins, we have to know that chromosomes are made up of DNA, but also histone proteins as well. Chromatin is when the DNA is unwound and loose during the interphase. A replicated chromosome is made up of two chromatids. That means that these single chromosomes have identical information, and they get held together by the centromere. And then one last little part that you need to know about the chromosome structure is the telomere, and those are the tips of the chromosomes. They wear off a little after each cell division, and they play a role in aging. Okay, so each time that tip wears off, then the a little bit of, you know, the chromosome, each time the cell divides, a little bit of that telomeres get wore off, and eventually that cell can't divide anymore. And so that really is what contributes to us aging. We don't live forever. So when you look at the chromosome structure, remember that DNA is made up of a double helix with all those bases. A pairs with T, C with G. Then those are wound around these histone proteins. That's the little purple things there. And then as that gets wound more and more, when it's still loose, it's called chromatin. But as it starts getting wound more and more and more, then it becomes a chromosome. And this right here is a duplicated or replicated chromosome. So the tips down here are the telomeres. You'd have those at the very ends. One of these right here, one of these is a chromatid, and this would be another chromatid. And how they're held together is by that centromere. And so you can see here, before that synthesis phase of interphase, you have one chromosome or one chromatid. This is one unreplicated chromosome. And then after we go through the synthesis phase of interphase, now it's a replicated chromosome right here. So you have two sister chromatids that are held together by a centromere. So here's one, here's one. And then when we go through cell division, those will get separated again. So here's an individual chromosome, and after replication, it's made up of two sister chromatids. Here's one, and here's one, and they're held together by that centromere. And here's another one, and here's another one. Because remember, you have two of every kind of chromosome. You get one from mom and one from dad. And so you'd have two of each of these types of chromosomes. So when we look at the whole cell cycle, remember, all of this right here is interphase. That's the first phase of the cell cycle. And so you have organelles being replicated and growth. Right here, you have DNA replication, and I think actually we're supposed to put the phases. I'm just describing because of what they've already written on the outside. But here, this is G1, or gap 1. This is the S phase, or synthesis. And here is G2, or gap 2. And again, there's more growth and preparation for cell division here. Synthesis, remember, is when your DNA gets replicated. Then the next phase is right here, from here to here. This is called mitosis. And it's made up of prophase, metaphase, anaphase. Sorry, you can't even read my writing. This is so tiny. I can't write on here. And telophase. And then finally, after that, we still have to physically divide the cell into two. Remember, mitosis is just dividing the nucleus. So we're just pulling those chromosomes apart. So half of them, one set of the chromosomes get to go in one cell, and one set of the chromosomes get to go in another. But we finally have to divide the cell, and that's cytokinesis. And that's when the cell physically divides, and now we have two cells that are the same. All right, let's look at chromosome structure then. Remember, the center part right here that holds two chromatids together is called a centromere. The tips of the chromosomes at both ends here are the telomeres. Okay, this right here is considered an unreplicated chromosome. Okay, and this right here is a replicated chromosome. Because here's 1 and here's 2. So this is a replicated chromosome, this is an unreplicated chromosome. Both of these are chromatids. In fact, they're called sister chromatids because they will have the same genetic information on them, because you took this one right here, this one right here, and you replicated it, so now it's into 2. All right? So this first one, again, deals with structure of your chromosome, and then you need to know the 3 parts of the cell cycle, as well as the 3 stages of interphase.